Section 2 and Video 2 of our Cell Molec Part 1 notes deals entirely with the parts of the eukaryotic cell. And so all the internal structures that are found within a eukaryotic cell are known as organelles. And it's really important that you know this term to start with. So when you see the word organelle, realize that that means one of these compartments inside of a eukaryotic cell. I have to be honest, it drives me nuts, or it has driven me nuts in the past, when I'll ask students for an organelle and they'll write something like plant cell or animal cell. It just kills me. Right, so you have to know from the get-go that an organelle is a structure inside of a plant cell or an animal cell. So let's take a look at all these things. And as we do, we're going to keep our focus on structure and function. So which tissues or which parts of your body would structurally have a lot of each particular organelle based on the job that it does for the cell. Alright, so we're going to start with the core organelle of the eukaryotic cell, and that is the nucleus. And so remember, the major thing that makes a cell a eukaryote is the presence of a nucleus. Now the nucleus we refer to oftentimes as the control center. This is where we're going to find your genetic material. It is surrounded by a double membrane referred to as the nuclear envelope or sometimes nuclear membrane. This has pores in it so that very small things can get in or out of the nucleus. But primarily we want to keep things in the nucleus. And that major thing that we're going to find inside of there is your DNA. Now also inside the nucleus, we find a region called the nucleolus. So notice this spelling difference here, nucleolus. The nucleolus is where you see a large amount of RNA, and the RNA that you see there is actually rRNA or ribosomal RNA. The nucleolus is where the cell puts together new ribosomes, which is another of the organelles that we'll cover here in a moment. You're also going to see the assembly of tRNA or transfer RNA in this nucleolus region. So here in our picture here, we've got three different cells. Here, if I outline them kind of in blue, and then inside that cell, you very clearly see a nucleus in each case. Now, as I said on the last slide, the nucleus contains the genetic information of the cell. Now, this is the DNA, and DNA is found wrapped around proteins called histones. A lot of times we say that it's wrapped around these protein spools. So in this picture, the red strip would be DNA, and you do not need to know the details of these different pieces here, but you wrap the DNA around a histone protein, like thread wrapped around a spool. Now, these histone proteins are then stacked together to store DNA as chromatin, and then the term that we're probably most familiar with as chromosomes. Now, before we can do mitosis, and I know we haven't covered that in this class, but I'm pretty confident you remember that term. Before we can do mitosis, we have to duplicate our DNA, or we have to copy our DNA. So to copy the DNA, we have to sort of unspool the DNA from these histones. Anytime really you're going to use DNA, you also have to unspool it a little bit so you can read that DNA sequence. But otherwise, the DNA is very tightly packaged with these histone proteins there in the nucleus. Now, different cells have different numbers of chromosomes depending on the species that you're looking at. Pretty much all eukaryotes have primarily cells that have an even number of chromosomes because those chromosomes appear in pairs. So, example here for humans, we have 46 chromosomes or 23 pairs of chromosomes. Now, a minute ago I mentioned that DNA is packaged by wrapping it around these histones. This is actually a really complex process, which allows a cell to be extremely tiny and still have tons of DNA inside of it. And we refer to this as chromatin packaging. So if we start out, we've got the DNA double helix, but you rarely see DNA by itself. Usually it will be wrapped around these histone proteins that we learned about on the last slide. Now, when you see DNA wrapped around a histone, we call that a nucleosome. So a nucleosome equals the DNA plus the histone protein. Now, once you wrap it around these spools and form nucleosomes, they will stack together to form what's called a chromatin fiber. So you see it's a big grouping of a bunch of these nucleosomes. Chromatin fibers are then kind of found tangled on themselves in sort of a scaffold. And that is what the cell looks like on a normal day. So generally when the cell is just sort of doing its thing, it's going to have this chromatin that's just kind of tangled up inside the nucleus. However, whenever mitosis begins, so whenever cell division begins, the chromatin will condense into a tightly packed structure known as a chromosome. And a lot of times when we see these under a microscope, they kind of have this X shape like you see here. This picture doesn't really do it justice, but what we're really seeing then is a tiny string, chromatin, wadded up real tight so that it looks much more dense. I like to relate this to if you've ever seen something maybe on the carpet and you thought, oh, it's a bug. And then when you walked over to it, it wasn't a bug. It was just a little string that was all wadded up. And so it looked like a little black ball, like a fly or something. But it was actually just a wadded up string. A chromosome is really just a wadded up string and that we see during mitosis. Alright, so the second organelle that we're going to take a look at here is the mitochondrion, or plural would be mitochondria. Now this is often referred to as the power center of the cell. Most eukaryotic cells in a multicellular organism like you and me contain hundreds of mitochondria. The mitochondria, kind of like the nucleus, also has a double membrane structure with somewhat of a more dense protective outer membrane and then an inner membrane that's really folded up like you see here. And these folds are referred to as cristae. Now, as we will learn here in, I don't know, a couple months, there are a bunch of reactions that have to take place on this membrane. So by having this highly folded membrane inside, it allows it to increase the surface area of that membrane. So you can think of this as though you took a big gallon-sized Ziploc bag and then wadded it up and put it inside of a little sandwich-size Ziploc bag. So that inner membrane is larger, it's just kind of folded up to fit inside. Now the area inside the inner membrane is known as the matrix, and the space between the two membranes is called the intermembrane space. Now to give you a little more detail of what we mean by the power center of the cell, the mitochondrion is the location where most of the cellular energy is generated. And cellular energy is generated through the reactions of aerobic respiration. Now aerobic means that it requires oxygen, so you're going to see this as kind of the site that requires oxygen in cells. And this involves the production of ATP, which is that cellular form of energy, via an enzyme called ATP synthase. So ATP synthase is an enzyme that helps you make ATP. Now here's our first structure-function relationship. Mitochondria are seen in all cells, but they're going to be really, really prevalent in tissues that need more energy. So for instance, if we look at a muscle cell, we're going to see more mitochondria per given area because that cell needs a lot of energy to produce force for your body. Now here's some interesting stuff. The matrix, the internal part of the mitochondrion, has its own ribosomes, its own DNA, and its own RNA. These mitochondria can also reproduce on their own without the whole cell reproducing. So mitochondria appear to be somewhat self-sustained. They can make their own proteins, they can divide when they want to, kind of makes us think that maybe mitochondria were at one time their own cell. So no one's for sure about that, but it leads to a pretty interesting theory about maybe where these mitochondria came from originally. Now, that theory is referred to as the endosymbiont theory, or sometimes the theory of endosymbiosis, is the other way you see this phrased. Now, this is a possible explanation for how eukaryotic cells evolved. What some scientists suggest is that an anaerobic cell, a cell that does not require oxygen, at one point engulfed an aerobic cell, a cell that did require oxygen. Now, the theory tells us that the larger anaerobic cell, which was prokaryotic, became the larger cell, and the smaller prokaryote that got engulfed became the mitochondrion. Now, this is also an explanation for the way that cells got chloroplasts, and also some would say a way that cells developed to have a nucleus. So basically, scientists suggest that some of the organelles maybe started out as their own cell, got engulfed or eaten by another cell, and you ended up with a more complex big cell that we now refer to as a eukaryote. So, pretty neat. Now, our next organelle is the ribosomes. And ribosomes are super important. Now, if you learn one big thing from this unit, it should probably be what ribosomes do. And we will come back later this year to look at the true function of ribosomes and how it works. But for now, it is imperative that you know that ribosomes make proteins. And now again, that'll be a huge part of our genetics unit later on down the line. But for now, you don't really have to know how they do it, but you got to know that they make proteins. So it's the major site of protein synthesis. Ribosomes are a solid structure. They're not a membrane enclosed structure like a bag. They're actually solid. And so this is the only organelle that we do find in eukaryotes and prokaryotes. So basically, this is the only organelle that prokaryotes have. Now, a ribosome itself is protein and RNA. It's formed from two subunits, a larger piece and a smaller piece. In a prokaryote, we say that it's a 70S ribosome, which contains a 50S large subunit and a 30S small subunit. I realize those numbers don't add up to 70, but that's just the way it works. 70S refers to its weight or its density. Now, this is a smaller ribosome than what we see in eukaryotes, which have a 60S large subunit, 40S small subunit, or an 80S ribosome altogether. So prokaryotes have smaller ribosomes, eukaryotes have larger ribosomes. And the ribosomes can be found freely floating out in the cell, which is what you see in this picture. This is actually a group of ribosomes working on a strand of RNA. Or the ribosomes can be attached to the rough endoplasmic reticulum, or rough ER. So this seems like a good time to talk about the endoplasmic reticulum, or the ER. Now, before we go too far, just realize that every cell has ribosomes, and every cell has lots of ribosomes, because every cell has to make proteins. But the more protein that cell has to make, maybe the more ribosomes it will have, but definitely the more endoplasmic reticulum and another structure, Golgi apparatus, that it will have. Now, the endoplasmic reticulum, as pictured here, is a tubular membrane system that actually starts as an offshoot of the nucleus. So notice it's actually connected to this nuclear envelope. The ER is a system of sacs, basically, that transports proteins and other products throughout the cell. So the inside of these sacs we call cisternae, and it is through these cisternae that the cell will actually sort of package and prepare and further lead to the sort of full development of proteins and other products that the cell generates. Now, there are two types of endoplasmic reticulum that you might remember, and you can see them here in this cartoon, and then also an actual real electron micrograph picture from a real cell here. You've got a rough ER and a smooth ER. Now, the rough ER looks rougher under the microscope, and it looks rougher because the rough ER is where you see ribosomes attached. Now, the rough ER is critical in packaging, altering, and finishing the production of proteins. So, associate rough ER with protein. And that's because the ribosomes attached to it build the proteins and then basically insert them into the rough ER. So, if you have a cell, here's another structure-function relationship, that has to secrete or release a lot of proteins, you're going to see a lot of rough ER in that cell. So the pancreas, as we learned with like insulin, releases a lot of proteins. Your digestive tract cells also release a lot of proteins that help you digest your foods. So these areas are going to have lots of rough ER. Now the other type of ER is the smooth ER. As you might guess, it is smoother. It does not have these ribosomes stuck to it, so it has a smoother appearance. Now the smooth ER has lots of jobs, but the main things is that it helps you process lipids or fats. It also helps you build hormones. Sometimes it stores calcium. Other times it releases glucose from areas like the liver and the muscles. Now, these are clues then of where you would find a lot of smooth ER. I often try to remember smooth ER deals with lipids. So think of lipids like oils as being slick or smooth, and so smooth ER. So if a gland makes a lot of hormones, something like the ovaries or testes, places where you see a lot of hormones being generated, that's going to have a lot of smooth ER because it's going to take a lot of fats and turn them into hormones. You're also going to see high amounts of the smooth ER in the liver and the muscles, which are where we have our glycogen stored carbohydrates that could be released as glucose when needed. All right, now after the ER, you have the Golgi apparatus. So this is sort of a further processing and shipping center. So if we were going to follow the path of a protein, all right, so here's my protein path. It's going to start at the ribosomes, pass to the rough ER, and from there pass on to this Golgi apparatus. And from the Golgi, it usually leaves the cell or goes wherever else it needs to go. So the Golgi is yet another set of membranous sacs. The inside of these sacs are also called cisternae. And the Golgi is, like it says here, essential in packing and transporting materials. It's sort of like UPS, right? So you build a material, the protein, the ER helps you get it packaged into a box, and then that box has to go through UPS or the Postal Service to get where it's supposed to go. So the Golgi receives materials from the ER. It modifies them usually by adding sugars, which is sort of the equivalent of putting like an address label on them, and then it sends those proteins out to wherever they need to go. Now the Golgi is going to package these materials into vesicles, which are just little pouches that will go around the cell to other organelles if that's where the protein should go. They might also merge with the cell membrane to release their contents outside of the cell, or the vesicle might just join the membrane to become part of the membrane. Now, something we saw on the last slide there is that the Golgi is also kind of close to the nucleus. So if we have our nucleus here, and then the ER comes off of that, and then the Golgi comes after that, we actually say that there's two sort of faces to the Golgi. There's a cis face, which is closer to the nucleus, and a trans face, which is further away. Now, most materials that pass through the Golgi move from cis to trans. Okay, so they come from the ER, go into the Golgi and then move further out to then be released. It's possible to see retrograde movement, where materials move backwards from trans to cis, but that's not as likely. So usually what we see is that materials go from cis to trans through that Golgi apparatus. Now here is an illustration of that. I just thought I would attempt to draw it myself first, see if it makes sense to you. But here's an illustration that came from a textbook. So the cis Golgi is closer to the nucleus, or ER. The trans Golgi is further away. Most materials are going to move in the anterograde direction, which is cis to trans, but things can move in that retrograde direction as well. So here's my super scientific term. Stuff, again, usually goes from cis to trans, but can also go from trans back to cis if it needs to. Now our next organelle is the lysosome. And the lysosome is a special vesicle or special sac that is responsible for intracellular digestion. Now, really important thing to take away right now. This does not mean food digestion. Okay, so a really common mistake that people make is they hear, oh, the lysosome does intracellular digestion, so digestive tract, foods, this is in the stomach. That's not true, okay? Lysosomes do intracellular digestion inside the cell. That just means breakdown, right? So what this really means is inside cell breakdown of whatever, right? Whatever stuff that needs to be broken down. There's a bunch of enzymes inside the lysosome that breaks down whatever the cell needs to get rid of. In the freshman year, we often refer to the lysosome as like the garbage man of the cell, because if there are extra cell parts or anything else, the Golgi will have those things sent to the lysosome, or they might just come into the cell and be sent to the lysosome, and the lysosome will engulf those pieces and will break them down. So this picture that you see here is actually a cell that is super duper active and has a ton of lysosomes here in yellow, which have been labeled for this image, where you can take in these cell pieces and you can break down that stuff that you don't need. Now the other interesting thing that the lysosome can do is it can fuse with the membrane and release its materials. So let's say we have a cell here, and we have a lysosome inside. Now that lysosome can fuse with or merge with that membrane. So we would end up with something that kind of looks like this. And when it does that, it's able to release those enzymes out of the cell. Now what could this do? This could send out these enzymes that break stuff down. So where do you see a lot of lysosomes? In the immune system. Because what we can do is we can use these lysosomes to help us destroy bacteria or viruses that enter your body. Those bacteria and viruses could get swallowed by the cell and broken down inside the lysosome, or we could spew our lysosomal enzymes out of the cell to destroy those bacteria floating around inside you. Now, one of our sort of medical ties in this unit is lysosomal storage disorders. Some people are born with genetic diseases which keep their lysosomes from working correctly. Basically, these people have a genetic problem to where they are missing an enzyme that should be inside their lysosome. And when you're missing that enzyme, you're not able to break down a particular product that you should be able to break down inside your lysosome. If you can't break those products down, what happens is they build up inside the cell, and the cell just becomes non-functional. Now, this is a really dumb analogy, but I think it gets the point across. You've probably seen hoarders before. Whether you've watched the TV show or not, you probably know what it is. So let's say you come to my house and I'm a hoarder, right? And normal people buy one box of cereal and then they eat it and they buy another box of cereal. But I decided that I was going to buy 12,000 boxes of cereal. Okay, and I'm going to put as many of those boxes of cereal as possible in my bathroom. So you come to my house and you need to use my restroom. And I'm like, sure, you can go in there. And you open the door to my bathroom and there's just floor to ceiling stacked boxes of cereal. You are not very effectively going to be able to use that toilet, nor would I very effectively be able to get a shower or anything like that in a bathroom that's that cluttered with junk. So a normal cell takes its lysosome and breaks down those things, eats the cereal, gets rid of the cereal. But a cell with a lysosomal storage disorder stores that material instead. And what it does is it keeps the cell from being able to do its normal functions. So just as you can't really use the toilet in a cluttered bathroom, you can't do whatever the cell's job is when the cell is totally cluttered with junk. Now there's over 30 different lysosomal storage disorders that are out there. These are really sad when you see them in young kids. But three of the more common lysosomal storage disorders are Tay-Sachs disease, eye cell disease, and Gaucher disease. And all three of these generally tend to store a product, I think in most cases it's a carbohydrate, in the nerve cells. And so in these kids, they start to lose some of their brain function, their other nervous functions, and then obviously this can lead to death. Now, one way that you can treat these types of disorders is called enzyme replacement therapy. You can't cure these diseases because, kind of like type 1 diabetes, it's genetic. We can't change their genes. But if you could provide them with the enzyme that their cell is not making, then potentially the enzyme would be enough to break down whatever that product is, kind of fix their lysosomes, and keep them alive for a longer period of time. Now our next organelle, and we're getting close to the end here, is the peroxisome. Peroxisome is kind of crazy looking. This is a computerized image, but the peroxisome is a neat organelle. It is oftentimes referred to as the epitome of compartmentalization, meaning it is a perfect example of why cells need to have little organelles. The peroxisome contains oxidative enzymes that will detoxify harmful substances within cells. Basically, these enzymes will combine with or will use oxygen to detoxify harmful substances in the cell. Now, the reason we call it a peroxisome is because when these reactions take place, these oxidative reactions, the cell actually makes hydrogen peroxide inside of the peroxisome. Now, peroxisomes are found at very high levels inside of any tissue that helps you to detoxify. So the liver is a primary place that a lot of people know that alcohol gets broken down. And so you're going to see a lot of these peroxisomes in that tissue because they help do that detoxifying job. Now, peroxisomes are an interesting case, however, because the fact of the matter is hydrogen peroxide is not good for you, right? Hydrogen peroxide is not good for you in your body there. So you're going to break things down in the peroxisome, but when you break down those harmful toxic substances like alcohol, you're also going to make another harmful substance, hydrogen peroxide. And so the cell also needs another enzyme that will break down that. Now, the other interesting thing about peroxisomes, as we said, this is a great example of compartmentalization. You have to keep these oxidative enzymes sequestered or kept kind of locked up inside the peroxisome. If those enzymes, which are oftentimes also referred to as oxidizing agents or oxygen-free radicals, were to get out of the peroxisome, rather than doing this nice little job of getting rid of toxic substances, those oxidizing agents could start to break down the cell itself. In fact, oxygen-free radicals have been shown to damage DNA and lead to cancer. So if your peroxisome were to leak some of its enzymes into the cell, it could be really damaging to the cell. But the fact of the matter is this actually does happen a little bit. And this is one of the major reasons why you may have heard of antioxidants in foods. Antioxidants are natural chemicals that are found in fruits and vegetables more than anything else. They are very high in grapes, so wine is actually high in antioxidants. It's also very high in tomatoes, so ketchup, Heinz ketchup advertises that it's very high in antioxidants. Any kind of fruits you eat, especially berries, if the fruit is red or darker in color, like a purple color, you're going to see a lot of antioxidants in there. And antioxidants remove oxygen-free radicals. And that's why antioxidants are good for you. Right? Oxygen-free radicals in your cells can cause cancer, can cause damage to the cell. So if you eat things that are rich in antioxidants, those antioxidants will remove those oxygen-free radicals and potentially help prevent cancer in you. So if you've ever heard people say before that like a glass of red wine a day is good for people, or obviously fruits and vegetables are good for people, part of that is because of antioxidants. So if you like berries, eat lots of berries. If you like ketchup, eat lots of ketchup. Now the last slide for this section is just an opportunity to quickly look at 3 different organelles found in eukaryotes but not found in animal cells. Now notice I'm not saying that these are necessarily found just in plants or anything, but these are organelles that we do not see in animals. The first of these is the chloroplast. Chloroplast is found in plants primarily. It gives those plants their green color. Chloroplast is the location of photosynthesis, okay, the photosynthetic reactions that we'll learn more about later on this year. And chloroplasts also have a double membrane, also have their own DNA, also have their own ribosomes, a lot like mitochondria. So a lot of scientists think that chloroplasts probably also came around because of the endosymbiont theory. Now the second organelle that we do not see in animals is the central vacuole. Central vacuole, also primarily seen in plants, this contains water, contains some enzymes, sometimes stores some carbohydrates, sometimes stores some wastes. But if you can think back to that picture of a plant cell that we started with, the central vacuole is usually huge. Usually takes up most of the area inside of a plant cell. Animal cells also have small vacuoles used for storage, but they don't have one big central one. So that's primarily only seen in plants. Now the last structure that we do not see ever in animals, but we see in several other different types of cells, is the cell wall. And the cell wall would be an extra rigid barrier outside the cell membrane, which gives the cell basically an extra protective coating. We see cell walls in plants. We also see cell walls in fungi. And if you think back to the previous video, we also see cell walls in prokaryotes. But basically in these different species, different types of organisms, the cell wall is made up of different components. In plants, it is made of cellulose, which you might recall was one of those major polysaccharides we learned in Unit 2, so a big structural carbohydrate. You're also going to see proteins. You're also going to see other carbohydrates as part of that cell wall. Fungi have unique cell walls. Prokaryotes have a structure that we'll learn about later this year called peptidoglycan. But for now, I really just want you to remember that plant cell walls contain cellulose. Alright, that wraps up Section 2 and Video 2 for Cell Molec Part 1, and we've got one more video to go.